Have you ever wondered why some people are more susceptible to certain diseases than others? Or why certain traits such as height or intelligence vary so widely between individuals? Well, today's episode is all about unlocking the secrets of the human genome through the revolutionary technique of genome-wide association studies, or GWAS for short. In today's episode, I sit down with Dr. Taliun, who is a computational geneticist, and his work is mainly focused on genome-wide association studies. Join us as we dive into the world of genetics and explore how GWAS is helping us unravel the complex web of genetic factors that shape who we are. All right, thank you so much again for showing up. This is actually your second time, but the first time in the new podcast. So I really appreciate it.
Thank you, Mike, for inviting me. It's my pleasure to be here.
My pleasure. I mean, not so many people do it once, let alone twice. So we can just dive into the questions. Yeah, so today we're talking about GWAS. So maybe we can start by a definition. What is GWAS?
Yeah, this is a very basic question, but it's also a hard one, especially explaining it to the broader audience.
Mm-hmm.
And recently I was actually giving a class to the students, introducing to them to GWAS, and when preparing to the class, I actually found at least 5 definitions different of genome-wide association studies. If, I can't give them all, and some of them are very technical, but the most important thing are 2 words in this. It's genome-wide and associations.
Right.
So I will start with genome-wide. Genetic studies can be different by their design and their goal, and it depends on the research question. And for example, many genetic studies study only one particular gene, for example, biology of which is known, but they're trying to understand more deeper what is happening when, for example, you damage this gene or mutate, introduce mutations to it, and so on. So those studies are usually called, candidate gene studies or single gene studies, and they're concentrated on one region, small region of our genome, on that particular gene they're interested in. Now, genome-wide tells us that genome-wide studies are actually not concentrated on a particular gene. They actually analyze entire genome. Doesn't matter if it is a gene. In fact, doesn't matter if it is like inside the gene, in non-coding part of the gene, or is it outside. And majority of results from such studies come actually from the genome which doesn't code any gene. So the genome-wide means that it's an entire genome, and I just can remind to our listeners that entire human genome is composed of around 3 billion base pairs, 3 billion, we can rephrase it as 3 billion letters A, C, G, and T. So genome-wide association studies are studying all of them at once. Now, the second part is association. And this word can mislead a lot of people, even a lot of researchers who are actually, in the field. Because when we say association, when we say the gene is associated with the disease, we don't necessarily mean that that gene is actually the main reason why disease is happening, or this gene is actually causing the disease. What we mean is that there is some link between the status of the disease and the gene presence between an individual. For example, assume you are a doctor and you have like hundreds of patients, and you have the access to their health information, the blood measurements, and it happens that you also actually sequenced their genomes, so you know all the letters in their DNA. And imagine that some of your colleague told you that, hey, at this specific position of the genome, I noticed that patients who carry letter A are more likely, more likely to have type 2 diabetes, for example.
Right.
And now you want to confirm this. And essentially what you do, you take all your hundreds of people, group them into those who carry the A in that position and those who don't. And then you look, what is the proportions of type 2 people with type 2 diabetes in one group and another. And for example, you find that in your particular case, you have 70% of those who carry A actually have type 2 diabetes. And among those who don't carry A, only 50% have type diabetes. Mm-hmm. Now, here is what happens. Can you actually say that it's causal? No, but you clearly that A causes that type 2 diabetes.
Yeah.
Clearly not at this stage. Because you don't know what environment those people live in. You didn't measure actually. You didn't test other nearby positions, and so on and so on. So you need to be very careful. Essentially, what you did a simple statistical association testing, and you can, for example, using a statistical method, you can derive a probability. What is the chance that you observed these frequencies, these proportions, just because you were lucky. Yes, and if this probability is very, very low, like below 0.00005%, then you say, yes, you definitely, not, you can't be so lucky, so definitely no by chance. Mm-hmm. Yes, so this is the idea, and that's what actually this means, that those, like coming back to the abbreviation, Genome-wide association study, GWAS, so they test every position in our genome for association with the disease. That's what it means. Now, I mentioned before that we have 3 billion positions in our genome on average, yes, but luckily for us, we don't need to test all of them. We know that the number of positions we actually different, it's not so large, although it's still big. It's around 3.5 to 4.5 million of positions where each of us can carry different letters.
Mm-hmm. Yeah, I really like what you said about the word association. So based on that, how do you ensure that the findings from a GWAS are accurate and reliable?
One word which would come into my mind immediately, it's replication. Mm-hmm. So going back to the example, so your colleague, when you're a doctor, your colleague gave you a hint, hey, I see those people carrying that A letter in that position, are more likely to be, there are more among them with type 2 diabetes. And then you actually didn't believe him immediately. Yes, I told that. Mm-hmm. You actually repeated this to your own patients. Yeah. And that it's an example of replication. Mm-hmm. Because otherwise you could have believed your colleague immediately and said, okay, I will start giving them, prescribe them, I don't know. Imagine it's sometime in the future, I prescribe them, CRISPR, some CRISPR-based, drug, yes, so to remove this mutation, but this would be quite wrong. So that what you want is basically to confirm the results by separate experiment. This is the first thing. And then another thing that when we talk specifically about genome-wide association studies, we test for association at each genetic position in your DNA, independently without looking around, for others. So we test independently, and for each genetic position we have this probability that we observe that association not by chance. And then there are multiple let's say many statistical methods how you can actually further refine your results to select out of those many positions you tested those which are more likely to be causal. And
Right.
at this point, you still don't say that they're causal. You're still speaking about associations, but here you end up with a list of most likely ones. Then probably the next step, you want to confirm by replicating it in the different genome-wide association studies. And there are many different designs, but the idea is that you basically repeat it and you should get a similar thing. Then to confirm, then probably you want to have some lab experiments, for example, on cell lines, yes, knocking out this, if this variant is inside if this position is inside the gene, maybe you want to mutate and see what happens to the cell line. Then after that, if things you observed with the cells make sense biologically, that you see that all the changes in the cell make sense in terms of what you observe in the human body, then probably you could proceed with the experiments on the animals, I imagine, yes. And this would be next step, and this involves again a lot of ethical considerations, regulations, and so on and so on. And then after that, you only can imagine trying to, at this point, you probably already understood how that variant you found, how that, letter which you observe in that position influence the life cycle of the cell, influence how the gene works. You understood the biology.
Right.
Yeah. And at this point, you can start speaking about, if you want to develop a drug, how you will use this information actually to develop the drug.
So the end goal is to develop a drug.
Yeah, this is the end goal of all the genetics is actually to develop drugs to cure diseases or prevent them.
Right.
Yeah.
So before moving on, I wanted to ask about your specific area of research within GWAS. So what are some of the projects that you're working on?
So maybe some of your listeners listened my first podcast, and I just repeat a little bit about my background. So by training, I'm a computer scientist.
Right.
And I started to work in the field of genome-wide association studies, starting from my master thesis. And there I actually tried to apply my knowledge in computer science, especially in optimizing the algorithms, computational algorithms, and for actually association studies. So in the beginning, I referred that there are 3 billion positions, and we do at 3.5 to 4.5 million positions we're different. But when you start having data, genetic data on many people, your size of the data you collected grows. This means that you need more storage. Yes, on your computer. It means that you need more time to run some algorithms. And some of the algorithms developed initially in the field of genome association studies actually can take like days, weeks, some of them even years, if you would scale them. If you just take algorithm developed like 20 years ago and try to run on the new data at the scale it is now, then probably you will wait 2 years to finish this computation. So it means that field required people and required some investment in developing these computational tools and optimizing them for the large scale data. And that's how that's what actually I started to do as a student, and I'm still continuing doing this as now as a researcher.
Yeah, perfect. So I think that would be a great excuse for us to talk about the computational methods, because what you just described, in my understanding, the whole thing wouldn't be possible without many technologies like cloud computing, software engineering, maybe data science and all of that. So how has all of that shaped and impacted the field of GWAS?
I would say that it relies a lot on the computational aspects and computational algorithms, and even when I say even to reproduce the results, to replicate results, people use different software, and they process the data in some specific ways, and one would think that just to develop efficient algorithms is enough, but no, you need actually to apply the best practices of the software development we know from like computer science or computer engineering. So you need to make sure that you develop software in the reproducible way, that you keep track of all your changes in the tool, and so on and so on. So the field really heavily relies on the best practices of software development. At least it is moving there. And if you go now and Google, for example, for the most commonly used software tools in the genetic association studies, in some analysis, you will find that the source code for these tools is open to everyone. It's located on the source code repositories like GitHub, or like specifically there are other software repositories which are specific to the field, but they are all under source control. They're all trackable, and anybody can look at them and fix things or propose their fixes, and very interactive in this sense. So, and this is important for reproducibility. Now, the second thing is efficiency. And as I mentioned, tools which were developed 20 years ago, if you run them like they are now, they will probably never finish running, at least in like decades. So, and the field was evolving together with the size of the data. So there are more and more tools efficient, and now even with efficient computational algorithms, you still need to make sure they run in the reasonable amount of time. You still want to parallelize them, yes, and you want to run them not on one CPU but on like hundreds probably. Then here you were speaking already about cloud computing, yes, and also storing these terabytes of genetic data also, and allowing many researchers around the world to use this data at the same time. We're again speaking about cloud computing because it's a good platform for also data sharing. That's why a lot of algorithms are developed to be able to run on this kind of platforms. But there is one nuance when we speak about genetic data on the cloud platforms, is about the privacy of the data.
Yes.
Now, our genetic data should be very confident. And here there are a lot of practices and a lot of monitoring happening with around the genetic data when it's stored on the compute clouds, especially on the commercial ones. So people want to make sure that the data is private, there is no leak of the data, and so on and so on. And but you can never be sure,
right?
You can never be sure. And so there are recent algorithms, and that's why in many genetic studies, when people actually participate, some of them have quite strict requirements about genetic data storage and movement. Some genetic data can't be moved at all from the institution to like commercial clouds. That's why there are new algorithms evolving which allow actually secure computations on the cloud when the data actually encrypted and it never decrypted. So the algorithms is able to run on decrypted data, produce results maybe a little bit approximated but with enough accuracy. So that's how probably in the future we will see more cloud-based algorithms which actually are able to work on encrypted data without moving or somehow endangering this data.
Right.
Yeah.
So I wanted to ask you, given your background, would you consider yourself more of a software developer or a data scientist?
I'm not sure. That's a good question. I think I already lost this this kind of boundary feeling. If you would ask me like this when I was doing my PhD, probably I would say I'm still computer scientist.
Right.
Now I don't know. And to be honest, more I work, more I meet people from very different backgrounds all over.
Yeah.
And essentially it really doesn't matter. I mean, the ability to work with people coming from many different fields is actually increasing the chance that you will do something good and better, because you have different points of view, you have different approaches, tackle the problem, you have different expertise, I think, and I already even, I don't know how to answer it, because majority of my work is basically data analysis now, but some part is still, development. It's hard to say, but I'm sure not up to date with the latest, techniques in computer science or things like that. Yeah.
So let's go back to the details of GWAS. Can you describe the process of conducting a GWAS from like the beginning to the end?
Yeah. Let's skip the first step then when you need to decide what is your actually question. Let's imagine you have already in mind we call it phenotype or disease, for example. Phenotype, you can study, for example, height. It's not really disease, but there are people with extreme height, small height, and in terms of evolution perspective, also interesting. So there are also, not necessarily disease, but imagine you want to study a disease. So the first thing you want to do is recruit the participants and do it's called phenotyping, but basically what you want to do, you want to measure all the parameters of the disease. It can be biological samples, blood you need for further for sequencing the DNA, but also you want some blood parameters now it's out of my mind, but you want other blood parameters. You want probably body measurements from the person, and you want actually to understand the diet of the patient. You want to provide ask couple of hundreds questions like what is a diet, what patients eats, how much it sleeps, and so on and so on. So you want to understand this and many other things, and it really depends on what you're studying. So, but the first thing is identify the disease of interest, identify what kind of measurements you want to take, questions to ask, and so on. Then the crucial part to decide how many participants do you want. Now, and my answer would be that you need a lot, especially nowadays when we see that you really need to characterize and understand and study quite rare variations in our genome, and rare means which appear in less than 1% of people. And to do this, you need to sequence a lot of people, because for every 100 sequenced collected participant, you get only one who will carry your let's say letter of interest. So, and that's how you need thousands of them probably. And it also will depend on the on how common is the disease. Common mean what is the prevalence, essentially, what is the percent of people with the disease in your population. Then you want to make sure that you account for the difference when you recruit patients or participants to a GWAS study. You want to make sure that it's a homogeneous group, or at least if you have multiple groups, that you have equal or large enough number of participants from each of the groups. As an example, depending on our culture, depending from where we come, we can have different diet. And this may impacts also lifestyle. And this may impact your, the way you interpret your findings also. And also we have differences in genetics as well. Some genetic variants are more common in Europeans, but less common in Africans, and other way around. So you want, to have, you want to have enough representation from each of the different, ethnic groups, if possible. Now, then you need to decide on the sequencing technology, yes, and this actually interconnected with your number of participants. Now, the most, if we're speaking about current situation, the best you could do is to do whole genome sequencing with a technology. We call it high depth, but it means that it's very precise in detecting rare variants. But now it's expensive. It can go up to from $800 to $1,000 per participant.
Right.
Now you're ending up spending like $1 million for sure, or several millions just to process this, just to read this DNA. Now on top of that, there will be a price to pay, to collect this samples, yes, and then these blood samples extract DNA. Then there will be price to pay to process it, yes, computationally. There will be many people involved. You need to hire bioinformaticians, you need to hire nurses, and so on and so on. So it accumulates a lot.
Yeah.
Now you can go with cheaper technologies but less precise, and actually will depend on your question, on the disease you study, and on your, maybe you have some prior knowledge about what you expect to find. Now, if you expect that there is no rare genetic variation involved in your disease, then maybe you can opt out from that expensive whole genome deep sequencing. Maybe you can instead just read a particular positions which are more common in the population and study only those ones, and the rest you can infer from the other whole genome sequencing data which is publicly available for research. So it's called imputation. So basically you kind of try to guess the missing values which you were not able to discover in your with actually the DNA sequencing machine, yes, or genotyping machine. If you know that somehow that there are only coding, only genes involved, then you can go with deep sequencing, but it sequences only the genes, and it saves you at least a couple of $100 per individual. So you can go with that, although you will miss all the regions which are not genes. Yeah. And there are several other options, but it's really a combination of decisions made by a researcher, and depending on the disease, depending on the question they have in mind, and depending how much money they have, they decide between how many participants to recruit, what technology to use. Yes. Now, okay, once you decided how many participants, what technology, you actually start your study. The participants, for example, you can do recruiting in the hospital, and participants will come to the hospital. They will fill in the forms, give their blood, do all the measurements. Now, I guess you need to wait until your data is ready for analysis. And it may take some time, half a year, even might be longer, depending again on the size of your study.
For genotyping?
Yeah, for sequencing or genotyping. So for sequencing might be even longer, because a lab needs to prepare the samples, to sequence them. Then bioinformaticians need to process this data, and so on and so on. So at the end, you will get, And probably you will need couple of iterations. For example, you plan to recruit like 20,000 participants. Probably you don't want to wait until all 20,000 come and fill in all these details about their disease and give their blood. You probably want to process like first 5,000, then 10,000, then the rest 5,000, and so on. So you have in waves. This can allow you to analyze your data before the study actually recruiting finishes.
Right.
Then once you have this, at that point you're ready to do your GWAS. And this basically involves just properly running your statistical tools which we developed. And these tools typically account for the relatedness between your participants. They account for presence of non-homogeneous groups, like of different ancestry groups, so that the findings you find in your GWAS not simply because of the differences in the historical differences of genetics or environment, but really because of that allele that letter is more often present in your disease, is people with disease than people with no disease. And then at this point, after you run this, you get for each your position, you get your p-values, or like probability that you observed this this association just by chance. Yes. And this probability should be very, very low. So it's I can tell you to our listeners, so but it's 5 times 10 in the power of minus 8. So it's 8 zeros after the dot.
Yeah.
So very it's very conservative. But even with such conservative threshold, basically this probability means that this is how likely you doing a mistake. You're falsely saying that this variant, this specific position, allele, letter in this specific position associated with the disease. So even with this kind of very conservative threshold, it's still possible that your discovery is false of association. That's where we can go back and remember what we were speaking about, replicating the findings and make sure that they're true findings. So that's why you would want to run this kind of association in the replication GWAS. So for this, you can when you're actually designing your GWAS study, you probably also, which I didn't mention, probably you want to think how you would replicate these results. It can be that you're already designing the study, knowing in advance that you will be replicating some findings, suggestive findings from other studies. So in this case, you kind of already replicating them, but probably you still want to think about how you will make sure that you will, disentangle this true associations from false. Yeah, you probably would think it might be that you can divide your study in the replication set, but there are many, many different designs, and I'm no way an expert to explain this. But in any way, so you have your probabilities that you don't observe that association by chance, and then you have probably, you might have a few hundreds of them, and then you need to distinguish which one of them are most likely causal. And then you end up actually running another set of computational tools. And these tools are often based on, some of them based on machine learning as well, where you actually try to prioritize those associations based on the, for example, strength of how much they influence phenotype, based on, for example, where they are located. You would believe more in the positions which are located in the coding part of the gene, rather those ones which are completely, a little bit outside of the gene. Yes. And things or which are in regions, are regulating the how the gene is expressed in the cell, things like that. So you try to prioritize, and eventually you will end up with your short list of the most likely causal variants, and even most likely genes which these variants are impacting. And from here you can think about designing another study with in the lab. Yes, for confirming with probably cells, what will happen if you knock them down or something like that.
So, what you just described, the whole process seems so expensive and labor intensive. Do we have any alternative for GWAS?
Yes, but it again depends on your research question. Remember when I tried to define, explain what GWAS is, I mentioned that there are studies, genetic studies, which are concentrated on one gene. That would be an alternative. But this is based on the fact that you have some prior evidence about the gene.
Right.
Other alternative, for rare diseases, very rare disease, is actually, it's a family-based genetic studies where you have a person who has very severe disease, and you sequence or investigate the DNA of this person and parents of this person. And then you see what you try to see if, if this person carries, so we have 2 copies of each gene, yes? And even if one of that copies is non-functional, another copy of the gene is still enough for a cell to survive and for your body to function properly. Now, what can happen is that if your parents carry 1 copy of such mutated gene each, but what you inherit, by chance, you were unlucky, inherited, those copies which carry the letters, which, and then you end up with a very rare condition. And this is, this kind of genetic study can, detect those. But, and the assumption is that because this condition is so severe, it can't happen, for hundreds of people or thousands of people in the general population. It happens only for like a few people, and it's so bad that it's basically destroys, destroys all the function of the gene. And that's how you can, you need to sequence only, close relative to understand which one actually, which one mutation, which was the mutation and which was the gene.
Yeah.
This was another, another type of genetic studies where you don't need actually to collect many participants, run this genome-wide association tests, or do this expensive high-depth sequencing on the DNA. Although for the family-based studies, you still do sequencing. Usually it's but the point is that you don't need to do for thousands of people, you just do for a few. And most often nowadays you do only, you sequence only the genes. Yeah, because the rationale is that the mutation is actually knocking out a gene. So, but there were some studies which were debating a little bit about, maybe nowadays it's better to actually do whole genome sequencing in that cases, because if you sequence only the gene, you may miss some mutations. For example, when we say that we sequence only gene, we mean we sequence only coding part. Right. But there is non-coding part called introns, which don't code any amino acid, but they still impact how the protein is actually concatenated once, amino acids are read, yes? So if the mutation is there, then it's also destroys the gene. So, but with traditional sequencing methods, which sequence only coding part of the gene, you don't get that. So that many people argue that, you probably want to do also whole genome sequencing for there. But again, it's expensive, but you don't need to do it on 1,000 people.
The advantage of GWAS over the alternatives is that it's hypothesis free.
Yes. Yeah. It's hypothesis free, and, it's more for like more complex traits, or we call it complex traits, such as type 2 diabetes, where many genes involved. Yes, there is no one gene where you just, there is one gene which we know involved, but it's not only one. There are many others.
So let's talk about some of the challenges of GWAS. What are some of the biggest challenges in conducting GWAS, and how have you addressed them in your work?
So if we speak about challenges directly related to my work, I would say there is still, computational challenges. And they are related to the size of the data, because as the field moves towards studying more changes in our DNA, which are quite rare, you need to collect hundreds of thousands of people, sequence them. So, and just to give you an idea, I think from top of my mind, to store a sequence data already aligned and so on, you need around 300 gigabytes per individual. Now, if you can, so it's 0.3 terabytes, so you can multiply, if you collect 100,000, it's like we are speaking about petabytes, so you need to analyze that. Here is my challenge, that, more end up with more efficient methods to analyze it. And another challenge is not about even efficient methods to analyze this or store this, but also help researchers to make sense of the results. Because final results are actually very multidimensional. So far we spoke about the setting where you are interested in one disease, and you kind of have, one disease only, you run one GWAS. But we know that, some of the genetic variations actually can be associated with many diseases. We call it pleiotropy. And again, I say associated, but not necessarily causal, okay? And then it means that instead of actually, it makes sense to look, kind of your logic tells you it makes sense to look not on, in this case, you want to look for other, other diseases with which that variant associated, or maybe other measurements, and see it does make sense or not. Okay, you have multidimensional results, and this is one example when you want to look. Another example is that nowadays we can access also medical records from people.
Right.
And it means that we have access not only to one measurement of one disease, we have access to hundreds and hundreds of variables collected when person visited the hospital. And imagine what will happen if you run a GWAS on each of those measurements, and you have you have millions of association statistics for each of the measurement. You have thousands of them, hundreds, and then you multiply and you get your number. And I just give you a hint. Mike actually tries to develop, as his part of his master thesis with me, he will develop a tool which actually will help researchers to visualize this data and make it browsable, and maybe it will help researchers to kind of develop some initial hypothesis about disease they want to study or they're studying. So this is another part, kind of trying to develop tools which help researchers to make sense of the results.
You mentioned the field is moving towards rare variants, right? So like overall, what is the future direction of the field? What are some of the new trends?
Yes, rare variants, so meaning more participants. We're speaking about millions at this stage. Second point is integration with medical records. We're speaking about thousands of measurements, of phenotypic measurements. Third point, integration with your portable devices.
Right.
For example, if you study genetics of diseases or conditions such as mental health, it's very difficult to measure.
Right.
And one of the ways to for example, you study genetics of sleep. One option is to invite person. There are some institutions have a special rooms for sleeping to study sleep. Basically, they invite you there to to sleep. It's kind of eliminates a lot of participants from your study. So what you can do, you can ask your participants to install on your smartphone app which will do your measurements, or use your Apple Watch measurements. Now we are talking about measurements from your portable devices, and we're talking about a lot of time points. And this is where it's moving.
So when you said medical records, how is it different from like basic questionnaires?
I would say it's more well, for the questionnaire, you need to be very careful. When you design a GWAS study, you are speaking about decade of research ahead. So you need to be very, very good at understanding what do you want to ask people, because they will come once and then you will need to deal with this data for next 10 years.
Yeah.
Now, you don't want to forget any question, and you want to be them precise. And that's end up in the sense. So, of course, there was many standard questionnaires developed. For example, if you're designing a nutritional questionnaire about nutrition of the people, you have already people developed for many years. This is a set of questions. But in any way, it's more static. You can go back in time or call participants back and hey, that question number 94. Or can I ask you some? It doesn't work, so it's static. You can come back. And for medical records, it's more precise, and it kind of you can go there and find things that are relevant for your disease you're studying, but about which you even didn't think of. For example, you designed a questionnaire for type 2 diabetes, but type 2 diabetes might be is quite kind of related to many other conditions, and you didn't think of asking about some specifics. But if you would have medical records for your participants, you can actually go and see, that makes sense. Those people, had this condition, they had these drugs. So you can, it's very, very unlikely that many participants will be able in your study to answer about which actually drugs they are prescribed to. Yes, when they fill in the questionnaire. I know, some studies were asking people to bring all the bottles of their prescription drugs to the recruitment center, so they can actually, with the help of nurse, they can actually list.
Yeah.
So, so not many people can do it, and there can be like many errors happening. But if you have access to medical records, you're more likely to get information in much better shape and much more precise. Did I answer?
I'm just thinking that doesn't it make any kind of a bias in recruitment? Because
yes,
not so many people will agree to do that.
Yeah, it makes a bias. Even without medical records, still there is a bias, because in recruitment, and it was shown in many population-based studies, and the bias is that it's social bias as well. People who are more willing to participate in genetic studies, they tend to be doing better economically, financially, and so on and so on. So from top of my head, I will not be able to list all possible biases, but there are biases even in the current even if you don't go with hospital-based recruitment with medical records, you still will have biases. The way you starting from the way you advertise your study, ending with the biases you can't control, that people who are more willing to participate have more opportunities to do this.
Yeah. Great. 2 more questions and then we can wrap it up. So what are some of the most interesting or unexpected findings that have emerged from GWAS research in recent years?
Good question. And I actually encourage you to ask this question to your other guests who work in the field or related field. So I in the beginning, I told that the main outcome from genetic studies, we expect drugs or new ways to treat people when they have some health condition. So I'm no way expert to talk about this, because I'm kind of work in the field where I kind of develop algorithms, and I'm not too much in the field of like final stages. But what was, I guess, interesting for me personally and for the field in general, apart the end product like drug development, is just understanding of underlying biology. So we still have a lot of lessons to learn. We're still not able to, even having technologies which allow to sequence completed DNA, still expensive, and we still don't have enough data to like understand the biology of variants, especially rare ones, and that they play a role in the disease. So I think the field is at the stage that we are convinced that it's very important to be inclusive in genetic association studies. That in there are differences between ethnic groups in how in their genetic variations and how they impact disease, and it's helpful to include everyone, because the findings are much more plausible and you have much higher chance to find something. I think I lost my track of I guess it's rare variants, the finding that we need to investigate more rare variants. That would be the main, and better phenotyping, better more medical records, more precise.
Right. Perfect. Thank you so much. Last question is, what advice would you give to young researchers who are interested in pursuing a career in GWAS research?
My advice, first, it's very, very interesting. Be ready to do a lot of hard work. And if you're into the computational things, if you like data analysis, if you like things like machine learning, it's a perfect field to apply this, actually, to polish your skills or obtain the skills or apply the skills, depending on what stage of your career of your career you are. So it's a very fast moving field, and it's I think it's in need of it's actually in the need of people who will work on this data and try to analyze it.
Thank you so much again. It was such a pleasure to have you on the show.
Thank you, Mike. My pleasure as always.
All right, I really hope you enjoyed this episode. If you did, please leave us a great review on your favorite podcasting platform, and I'll see you again next time.